This meeting is now being recorded.
The presenter for today is Dr. Fanny Tang. Fanny holds a PhD in nutritional biochemistry from the University of North Carolina at Chapel Hill. She joined Metabolon in 2014 and is Senior Study Director, where she's responsible for analyzing and interpreting large metabolomic data coming from various industries with a focus on applied markets, including human and animal health and nutrition, consumer goods, and skincare. I'll be turning the presentation over to Dr. Tang now.
Thank you for the introduction. It is great to be here with you today to talk about metabolomics and microbiome. They have different levels of experience or exposure to metabolomics. With that in mind, we will start the webinar with a short primer of Metabolome and metabolomics. We will then talk about the utility of this technology in host and microbiota research, and this is exemplified by past case studies. So Metabolome measures small metabolites. Those are not protein, DNA, or RNA, but small molecules that comprise metabolism. While changes in DNA, RNA, and protein may never have the penetrance to impact the physiological state, metabolites are the closest to the phenotype. They are very sensitive to changes and can be influenced by factors such as diet, environment, and of course microbiome. So really, the measurements of those small metabolites serve as a summation of all the factors that drive the phenotype and provide a readout of the physiological state. In addition, those metabolites are also actionable, meaning that the levels may be potentially intervened by treatment. And that's what we are interested in. So the measurement of small metabolites take place in our regular life. If you go to a clinic, the doctor may take the blood sample to measure glucose or creatinine to assess insulin or kidney function. Those are all examples of small metabolites measurement. However, while most settings can only measure a few metabolites from one given sample, Metabolome tries to provide you the broadest coverage, and we can report 100 to 1,000 metabolites from one given sample. So to put this visually, instead of providing you a street view, we want to provide you a view of a whole map and hopefully also the direction that drives your research forward. So this is the screenshot or the wall chart of all the small metabolites that circulate in our body. And each of the blue dots represents a metabolite we can detect in human plasma samples just to show you our coverage. And essentially what Metabolome does is to take the samples from the client, and it can be biological fluid, cells, tissues, any matrix that you can probably think of. We probably have worked with it before. And what we do is we screen across all of those metabolites, and not only we detect the metabolites that you may be looking for example, the TCA cyclointermediates, but also metabolites in pathways that you may previously not anticipate. So by using this unbiased global approach, we provide new insights to your research question. The challenges of wanting to detect all of those metabolites is that those biochemicals have very different properties. And so over the years, we have established our assay to employ 4 different methods. All of those are mass spectrometry-based methods. And each of the platforms generate large amount of ion features. Those hundreds and thousands of ion features do not represent there are hundreds and thousands of metabolites. Those are fragments of metabolites. So there's really a lot of noise in this data set. Other groups may take a more ion-centric approach, meaning that they may take those hundreds and thousands of ion features and perform statistical analysis. And based on the false discovery rate, if you do start at hundreds and thousands of ion features, you will probably see about 10,000 ion features that are significantly different between the experimental, the test and the control group. Then we need to figure out what those 10,000 ion feature means. And Metabolome does things very differently. So we take what's called the chemocentric or library-based approach. In our library, we have about 4,000 named biochemicals, and when those were added into the library, they were run with purified standards, and each biochemical has very unique signals, including retention time, actual mass, and fragmentation pattern. So by query those specific parameters coming from the client samples, those in our library, we can get really accurate determination of each metabolite. And in fact, in the world of metabolomics, the uses of 3 orthogonal signals to identify a metabolite is considered tier 1 identification, while other groups may only use 1 or 2 of those orthogonal signals. So once the list is generated, we then perform statistical analysis, and we understand what those metabolites do. We can classify them into super and sub pathways, and we can start understand what metabolites or what pathways have been impacted by the experimental conditions. We then perform interpretation, and over the years, we have completed more than 5,000 studies, and work done between us and our collaborators has led to more than 700 publications. So there is really a lot of knowledge coming out from our work. So this is a very quick description of what we do. Now, the next question is, why is this important for host and microbiota research? Microbes, just like humans, produce a lot of metabolites as well, and they use those as the currency or the language to communicate with each other or with other species like plants and humans. In fact, in our body, there are probably trillions of microbes that live in our GI tract, on our skin, on other mucosal surface of the body. And they play really essential roles. Those microbes can digest the food we cannot digest. They can synthesize vitamins that we need, or they can prevent the colonization of unwanted pathogens. And those small metabolites produced by those microbes have may have signaling roles as well. For example, the production of serotonin, and serotonin is a neurotransmitter that is in our body, and that is thought to be responsible for the feelings of happiness. And interestingly, only 10% of serotonin is produced in the brain, and 90% of the serotonin actually comes from the GI tract. And this study that was published 2 years ago in Cell, and also were partially contributed by Metabolome, has found that microbes can produce small metabolites, specifically PABA and tyramine, and those small metabolites can act as signaling molecules to stimulate serotonin production by the host, leading to increased serotonin level in the column as well as in the serum. Small metabolites produced by those microbes also can be involved in the health status of the host. And one example is work that's done by Dr. Stanley Hazen from Cleveland Clinic, where he shows that microbes can take nutrients such as choline and carnitine, those are really abundant in red meat, and convert those to the metabolite TMA, trimethylamine. And our liver can further convert trimethylamine to trimethylamine N-oxide, which is the metabolite that is associated with increased cardiovascular disease risk. So the examples that showcase the importance of those microbial-derived metabolites can go on and on and on. And however, despite the great interest in metabolomics, many microbiome researchers seem uncertain about the quality that a metabolomics technology needs to possess in order to illuminate the functional aspects of host and microbiota interaction. While the major workflow right now focuses a lot on just a small portion of metabolites, and if I ask you what metabolites you may want to see when it comes to microbiome research, you may probably think about short-chain fatty acids. There is nothing wrong with short-chain fatty acids, but by taking a more targeted approach, it really limited the capacity to decipher the biological story. So when we consider a technology that is needed to understand the influence of the microbiota in health, we think there are four major classes of metabolite that need to be systematically measured. Of course, the technology needs to be able to pick up established microbiome metabolites, but the technology should still also able to have the capacity to pick up novel microbial-derived metabolites, dietary metabolites, drug metabolites, and host metabolites, so that we can start understand how the host interact with those microbes. And we have done this pretty successfully. In our library, we probably have about 200 metabolites that we know are exclusively or predominantly coming from bacteria. And those include a lot of those secondary bile acids, metabolites derived from aromatic amino acids. In addition to those established microbiome metabolites, from one given sample, we can also provide information on amino acids, energy, lipid metabolism. So really, this unbiased global approach is really essential for us to understand how the host and microbiota interact with each other in order to result in a very specific trait or phenotype. So in the next half of the presentation, I want to give you two examples of how other researchers have applied this technology to their microbiome research. And the first example I want to give you is related to this yo-yo effect. Many individuals fail to maintain weight loss. Instead, they enter this cycle of repeated weight gain and weight loss, known as the yo-yo effect. To better understand the mechanism driving this effect, the investigator turned into a cycling high-fat diet model in which the animal were fed high-fat diet interleaved with normal chow. Animals fed the normal chow, high-fat diet, over one cycle of high-fat diet served as the controls. As expected, animals that fed the normal chow gained the least weight. Animals fed the high-fat diet gained the most weight. Interestingly, animals that were exposed to 2 cycles of high-fat diet gained more weight than the animal that exposed to 1 cycle of high-fat diet, even though their weight has returned to normality after the first cycle. So based on this information, this data, the investigator hypothesized that the preceding weight gain and loss cycle has predisposed the animal to accelerate weight gain after refeeding high-fat diet. And so they turn into metabolomics and microbiome profiling. On the left-hand side here, we are looking at the data from 16S ribosomal RNA sequencing, and basically those microbes are clustered based on their response, the similarity in the response to obesity. And during obesity, there is a dramatic shift in microbial profile that many microbes were either lost or gained. Once the animal lose weight, some of those changes were reversed back to the pre-obesity state, but many other microbial-derived changes persist weeks after obesity. And the same thing can be said with the metabolomic profile, that obesity caused a dramatic change in those metabolites. Some metabolites were lost, some metabolites were increased. While many of those metabolites can be reversed back to the pre-obesity state once after dieting, many of those metabolite changes persist even after the weight has returned to normality. Among those metabolites that persistently altered are flavonoids, apigenin, and neurigenin, and their level were reduced during obesity and continue being suppressed up to 15 weeks after obesity. And if you think about this, 15 weeks of mice age is equivalent to 26 years of human age. So this is not a short-term effect. And those flavonoids are coming from the diet and metabolized by microbes. So as it turns out that during obesity, there is a shift in microbial composition to favor flavonoid degradation, resulting in the reduced level of dose. And the changes in microbial composition as well as the flavonoids degradation persist even weeks after dieting. So to better understand the involvement of flavonoids in this cycle of weight regain, the investigators turned into the next experiment, and that is to compare flavonoid supplemented cycling high-fat diet group to the control group that does not have flavonoid added. And they showed that by reintroducing flavonoids back to the diet, this flavonoid is able to ameliorate the secondary weight gain. The investigator also showed that during obesity, the cycling high-fat diet has reduced energy expenditure, and this reduced energy expenditure was reversed with the supplementation of flavonoids. So this is a paper with a lot of data, and I really encourage you to read the article to find out more detailed information. But in summary, the investigator was able to show that during high-fat diet, there is a dramatic shift in bacterial composition to favor flavonoids degradation. And since flavonoids is involved in energy expenditure, reduced level of flavonoids led to reduced energy expenditure, therefore accelerated weight gain upon refeeding of high-fat diet. And this is an example that if the investigator took a more targeted approach and only measured microbial-derived metabolites, they may miss out the opportunity to detect those flavonoids that is essential for their study. The next question is related, the next example is related to the disease autism spectrum disorder that is characterized by stereotypic abnormality and deficits in language and behavior. Interestingly, individuals with autism tend to have higher prevalence of GI abnormality, leading to the proposal that the gut microbiota may play a role here. So to test this hypothesis, the investigator turned into a maternal immune activation mouse model. Basically, in this model, the pregnant dam were injected with polysaccharides infection, and the offspring coming from those mothers have autism-like behavior. And this is related or based on the epidemiology studies linking maternal infection to increased risk of autism in the offspring. On the left-hand side here, we're looking at FITC intensity, which is a measurement of gut permeability, and DSS is our model of colitis. So as you can see, the positive control model of colitis have very high level of gut permeability. The offspring coming from the infected mother have higher FITC level compared to the saline control, suggesting that they do have leakier gut. Importantly, those changes not only were seen in adults, but also in 3 weeks of age, suggesting that this abnormality takes place really early on in age. The investigator also measured microbiome profiling, and here we are looking at the principal component analysis of the operational tox units. And as you can see, the samples coming from the infected mother is separated from the saline control, suggesting that the two groups indeed have very different microbial composition. The animal also showed autism-like behavior, and one of the tests that's used to test stereotype behavior in rodent model is the marble bearing experiment. And it is based on the observation that mice like to bury objects beneath the bedding. And the more they bury, the more OCD behavior they're showing. And the offspring coming from the infected mother bury more marble compared to the saline control, suggesting that they have more of this stereotype behavior. And this behavior is reversed when the gut permeability is treated or ameliorated with B. fragilis. So those data suggest that there is indeed a link between gut bacteria and autism-like behavior in rodents. Now the next question is, what is the missing link? So they turned into Metabolomics and sent us the serum sample From the serum sample, although we detect about a few hundreds of metabolites for this study, and out of the hundreds of metabolites that we screened, only 8% of metabolites were significantly different between the two groups, which is not a large number. However, among those that changed, this specific metabolite 4-ethylphenol sulfate, the level was drastically elevated in the poly-IC offspring. The level was increased by almost 50-fold compared to the control. And once again, this was reversed when gut permeability was ameliorated with the B. fragilis treatment. And this metabolite is microbial-derived metabolite and synthesized from the amino acid tyrosine. Importantly, this metabolite also shares a similar structure with another metabolite for p-cresosulfate, which is a putative biomarker in humans with autism. Of course, the next question is whether 4-EPS can induce this anxiety-like behavior. So what we're showing here is another behavior testing. It's called the open field exploration test. The mice are allowed to spend a certain time in the open field, and the distance travel and the time they spend in the center or outside the center have been recorded. Animals that spend less time in the center are reflective of more anxiety-like behavior. And offspring coming from the infected mother spend less time in the center compared to the saline control, and this was reversed with B. fragilis treatment when gut permeability was ameliorated. Importantly, the investigator was able to show that by just taking the wild-type animal and daily injection of 4-EPS, it is sufficient to induce this anxiety-like behavior. So this is another example that metabolomics is used to understand the host and microbiota interaction. And out of the hundreds of metabolites that we detect, we were able to narrow it down to specific target and specific biomarker. So I just want to close by saying that a lot of investigators think about applying metabolomics to their research. They often only thinking about the fecal sample, which makes sense because a lot of the 16S signature is based on the fecal samples. However, we do have a lot of experience in other matrices such as plasma, serum, urine, and skin, and we also see a lot of microbial-derived metabolite in those matrices as well. So if you don't remember anything that I say today, perhaps just remember one thing, and that is do not limit yourself. Do not limit yourself by only thinking about fecal sample or only thinking about a few metabolites. This unbiased global approach, this technology can really bring you a lot of insights. Thank you for your time today.
We'll be answering questions now in the order that they've been received. In the event that we run out of time, we'll answer your question offline as soon as possible. I'll be unmuting the mics. If you're not asking a question, please mute your mic to avoid any kind of feedback.
All guests have been unmuted.
So the first question is, what are the remaining challenges in the analytical approach taken by Metabolon? Well, while Metabolon is successful in doing metabolomics profiling, I think there are still many remaining challenges, and we can probably do a full webinar on that topic. The short answer is, how do we make this technology better, faster, and cheaper? We're constantly improving our process, so can we see more about chemicals without increasing the runtime time? We're looking for approaches to speed up the curation process, so maybe faster. And if we can make everything faster, then we may be potentially cut down the cost as well. So those are kind of the questions that we have in mind. And another biggest question is, can we do what we do on dry blood? And if it works, then the investigator may not need to centrifuge the plasma or freeze the samples. And, then this technology can be made available to a wide community. So those are all of the questions and the challenges that we would like to address in the future. Another question is, address any application on the use of stable isotopes for metabolic pathways related to microbiome. And another person also asked about lipomics. So because of the short time of the webinar, what I have talked about today focused on the global platform for the small metabolites. And as I've mentioned, the purpose of the platform is to screen as many biochemicals as possible, and those metabolites are identified based on accurate mass retention time fragmentation pattern. So this process, the global platform does not involve stable isotopes. I do want to mention that we do have a separate targeted assay team which use isotopically labeled internal standards to measure metabolites of interest, and they can generate standard curves and use it to calculate the absolute concentration. So if you have metabolites of interest, we can certainly work with you. Our target assay group can certainly work with you to establish the assay for you. In terms of lipomics, we do have a complex lipid platform in addition to the small metabolite HD4 platform that I mentioned today. And in the complex lipid platform, we monitor about 15 classes of complex, including phospholipids, sphingolipids, neutral lipids, and also the fatty acid species within the classes. So some investigators may find this additional complex lipid platform to be useful for their research, and we can certainly do those as well. Another question is, can we detect short-chain fatty acid levels in a way that they accurately represent their levels in the column? We do have a targeted short-chain fatty acid panel that can report the level of short-chain fatty acid in absolute concentration, so we can certainly measure those. I'm not entirely clear about the question, but my guess is that you're getting into whether the short-chain fatty acid in other matrices like plasma or fecal samples can represent their levels in the column. I'm not sure we can make the short-chain fatty acid in other matrices to represent the levels in the column, but my guess is that the more proximal of a matrix to the column, the data may look closer. So I would expect that the short-chain fatty acid in the fecal sample may be closer to the level in the column as compared to the plasma sample. The next question is, does Metabolome have some way to determine what blood metabolites are likely contributed by bacteria? And yes, we do. So as I have mentioned before, in our library, we have about 200 biochemicals that we know are exclusively coming from the bacteria. And we can also detect a lot of metabolites that are coming from the host, or it can come from both host and bacteria. The reason that we know those metabolites are specifically coming from bacteria is because some is based on the literature, some are based on the experience. So we have done several studies where we look at the metabolic profile from germ-free model compared to mouse models that are colonized with germs. And by comparing the metabolic profile, we can know which metabolites are missing from the germ-free model. So we can, that's one way for us to determine, yes, we know that this bacteria is, this metabolite is contributed by bacteria. So yes, we do have ways to determine the metabolite that's coming from bacteria, even if it's in the blood samples. And another question is, are those metabolites translatable? Since metabolism is central to all kingdoms of life, those pathways are really highly conserved and really translatable across species. So we know that the function in glucose is probably the same in human and mouse. And examples after example, we have observed striking translatability. For example, reduced choline can contribute to fatty liver in most human and mice. So we think that those metabolites are definitely translatable from species to species. You have discussed metabolites measured on the Discovery HD4. Do metabolites coming from bacteria on the Lipidomics platform? So I think there are some metabolites from the lipidomic platform that may be contributed by the bacteria as well. If we do especially for example, if we do some of the FAME analysis on the skin, some of those branched-chain amino acids on the skin are derived from bacteria as well. So I think that the bacterial contribution is not limited to just the small metabolites, but some of those lipids, especially the branched-chain lipids, are coming from bacteria as well.
Attending our webinar. In case you'd like to share the replay or listen again, the recording will be available soon on our website at www.metabolon.com. If you've submitted a question and we haven't had an answer, we will answer your question offline shortly. Thank you.